video is all about my immediate learnings in hypersensitivity and complement. Complement system, how is it activated? There are three pathways in the complement system: the classical pathway, alternative pathway, and the lectin pathway. For the classical pathway, the main trigger of the classical pathway is the antigen-antibody reactions. Activates C1, cleaves into C4, C4 cleaves into C4A and C4B, then C1 combined for C4B result to C1,4B. C1,4B enzyme converts C2 into C2A and C2B. C1,4B and C2A, these two complements combined and produce C1,4B2A, this enzyme known as C3 convertase. The C3 convertase splits C3 into C3A and C3B. The C3B combines with the C3 convertase forms C1B2A 3B. This is also an enzyme known as C5 convertase. The C5 convertase will now convert C5A and C5B, wherein C5A go to the circulation, act as anaphylatoxins. And C5B for the membrane attack complex, C5B6, 7, 8, the slow lysis, and the C5B6, 7, 8, 9 for the fast lysis, and product is the lysis. For the alternative pathway, the antibody-antigen reaction will trigger the activation of classical pathway, but in the alternative pathway, it is triggered by bacteria and the toxin, yeast cell wall, and the alternative pathway begins with the activation of factor C3. Present in normal serum, factor C3 converted into factor C3b allow the process of factor B to C3b. And open the active site of factor B, factor D, another complement enzyme split up factor B into subcomplement BA and B, big letter B and small letter B. Factor BB combines with the factor C3b, produce factor C3BB and forms C3 convertase, converts C3 into C3A and C3B. Same with the classical pathway. And that is the formation of the C5 convertase. And subsequently, the formation of the membrane attack complex with the formula C5B6789. Now let's talk about the lectin-mannose pathway, known as mannose binding lectin synthesized in the liver. Mannose binding lectin is the part of bacteria or bacterial cell wall, the mannose and the glucose, recognizing those and activate protease called MASP or mannose binding lectin associated serine protease. Is a protease which binds mannose binding lectin. MASP triggers the activation of C4 and C2 into their subcomplements C4A and C4B, and similarly C2 into C2A and C2B. The factor C4B and C2A combines the C4B, C2A or the C3 convertase of the lectin pathway. The C3B combines with the C3 convertase forms C1,4B2A 3B. This is also an enzyme known as C5 convertase. There are different immune effects of the complement products. First is the opsonization or the C3b. Second is the cell lysis or the membrane attack complex effect of C5b6, 7, 8, and 9. Has unruptures of bacterial cell wall. The third one is the chemotaxis. C5a attracts to to neutrophils and macrophages to the area where antigen is present. The last one is the activation of the mast cells. The factor C3A, C4A, and the C5A have a direct effect of the mast cells and the basophils. And this causes mediators of inflammation. The C4A, C3A, and the C5A is anaphylatoxins. Let's talk about the hypersensitivity reaction. Hypersensitivity reaction is an appropriate immune response to benign antigen. The drug, pollen, and the food can elicit inappropriate immune response. Hypersensitivity reaction can be the mechanism of diseases. In some order of reaction, hypersensitivity reactions are antigen specific. There are 4 types of hypersensitivity reaction. Type 1 to 3 are antibody-mediated, wherein the type 4 is cell-mediated. Hypersensitivity type 1 or the immediate reaction, example is the allergic reaction, and really involves of an antigen by primed IgE. Lymph nodes is sensitization occurs in initial exposure to particular antigen. Type 1 hypersensitivity reaction may cause from minor inconvenience to death. Reaction takes 15 to 30 minutes from the time exposure to the antigen. Diagnostic is the ELISA. Type 2 hypersensitivity known as cytotoxic hypersensitivity and may affect variety of organ and tissue. Reaction time is minutes to hours. Primarily mediated by the antibody of IgM and IgG. Diagnostic is the biopsy by immunofluorescence. Type 3 hypersensitivity known as immune complex hypersensitivity. Reaction may be general or involve individual organs including skin, blood vessels, joint, etc. Reaction will be caused by microorganisms 3 to 10 hours after exposure sa antigen. Mostly of IgG and IgM. Diagnostic is the immunofluorescence, but it is granular. The last one is the type 4 hypersensitivity, known as cell-mediated, involved pathogenesis of autoimmune and infectious diseases. Granulomatous due to the infection. Diagnostic tests in vivo include delayed cutaneous reaction, Mantoux test, and patch test. In vitro tests include mitogenic response, lymphocytotoxicity, interleukin-2 production. That's all. Thank you.